Epidemiological studies have shown that increased physical activity is associated with up to a 30% reduction in the risk of developing endometrial cancer. Physical inactivity and obesity are well-documented risk factors with the development of endometrial cancer. This can be attributed to significant increases in exposure of the endometrium to estrogen generated by fatty tissue deposits. Several studies have shown obesity increases the risk of endometrial cancer by a fourfold, and a sedentary life can increase the risk by up to 46%. A study completed in 2011 found while overweight women had a 1.5 times higher risk of developing endometrial cancer compared to women at a healthy weight, obese women had almost 5 times a normal weight woman's risk of developing the disease. Importantly, women who experienced a 35% weight gain in their 20s developed endometrial cancer approximately 10 years earlier compared to women without such weight changes early in their life. Achieving a healthy weight may be one of the most impactful lifestyle changes that women can make to reduce the endometrial cancer risk and potentially improve the treatment outcome. Although type 2 diabetic women have for many decades been observed to have a higher risk for developing endometrial cancer, the actual contribution of insulin resistance and diabetes towards developing the endometrial cancer has only recently been appreciated. Since diabetes typically leads to weight gain, and because fat deposits in overweight and obese people secrete estrogen, and this promotes endometrial cancer development, it was thought the contribution of diabetes to endometrial cancer risk was only indirect. It's now understood that diabetic, non-obese women who are otherwise healthy continue to be at a higher risk for developing endometrial cancer. This information is supported by the fact that metformin, an anti-diabetic agent, appears to hold considerable promise in the prevention of endometrial cancer. The traditional dietary pattern of populations of the Mediterranean region has come to be regarded as one of the healthiest eating styles in the world. Centering upon whole grains, vegetables, fruits, olive oil, fish, moderate dairy and wine, the Mediterranean diet has been shown in a large body of published scientific literature to reduce the risk of several of today's most prominent ailments, including obesity, cardiovascular disease, and cancer. Accordingly, rates of endometrial cancer in the Mediterranean region are lower than in the United States and the United Kingdom, and this discrepancy is thought to be due, at least in part, to the differences in the eating styles between these regions. In fact, it's been estimated that 10% of endometrial cancer cases could be prevented if Western societies shifted to a Mediterranean diet. In one study, adherence to a Western diet, which is high in saturated and animal fats, as well as refined carbohydrates, was associated with a 60% increased risk of endometrial cancer. Carotenoids are a family of yellow pigments and they're found in plants. One of the most prominent carotenoids, beta-carotene, is converted to active vitamin A within the body. Vitamin A and its derivatives bind to activate specialized receptors, and these contribute to regulating a process called transcription, which is the reading of the information encoded within the DNA. The activation of these receptors exerts several chemopreventative effects, including the inhibition of carcinogenesis, the induction of tumor cell death or apoptosis, and the suppression of tumor growth and invasion. Greater consumption of vitamin A or beta-carotene has been associated with a lower risk for developing endometrial cancer. In one analysis of dietary factors associated with endometrial cancer, the greater consumption of beta-carotene, along with vitamin C, was associated with a 50% reduced risk of the disease. Vitamin C, also referred to as ascorbic acid, is associated with a significantly lower risk for developing endometrial cancer. Vitamin C has been proposed to reduce the activity of a key protein called hypoxia-inducible factor 1-alpha, or HIF-1-alpha, and this is involved in endometrial tumor cell survival. In addition to its direct inhibitory effect on the tumor cells, the vitamin C was also proposed to boost the anti-tumor immunity. Specifically, it's also been suggested that vitamin C can aid the immune system's surveillance of the tumour cells and promote tumour cell killing. Several studies have shown the consumption of foods rich in vitamin C are associated with not only significant reductions in the endometrial cancer incidence, but also the disease grade. For example, one study showed the level of 50 milligrams per 1,000 calories consumed of vitamin C reduced the risk of endometrial cancer by 15%. Another study showed the highest quintile of vitamin C intake from the food, which was defined as greater than 72 milligrams of vitamin C per 1,000 calories per day, was associated with a 20% reduced risk of endometrial cancer compared with the lowest quintile of intake, which was defined as less than 29 milligrams per 1,000 calories per day. Consumption of foods rich in vitamin E are associated with a significant decreased risk of developing endometrial cancer. Wheat germ oil is very high in natural vitamin E. Nuts like almonds and hazelnuts are moderately high in vitamin E, and tomatoes and spinach contain lower levels of vitamin E. In one study, the highest intake of dietary vitamin E was associated with a 56% reduced risk of endometrial cancer compared with the lowest intake level. Naturally occurring vitamin E exists in 8 chemical forms: alpha, beta, gamma, and delta tocopherol, and alpha, beta, gamma, and delta tocotrienol, and these have varying levels of biological activity. The gamma tocopherol has been shown to possess significant anti-inflammatory and anti-tumor effects in a rat model of breast cancer. Of interest in the context of endometrial cancer, the anti-tumor effect of the gamma tocopherol appeared to be dependent on inhibiting the activity of estrogen. Given that endometrial cancer can be driven by excess estrogen or imbalances in estrogen and progesterone levels, it's tempting to speculate that gamma tocopherol can also have therapeutic activities against endometrial cancer, though studies are needed to explore this possibility. However, evidence has shown gamma tocopherol consumption might reduce the risk of gynecological cancer. A study conducted in Korea found that women who consume the highest levels of gamma tocopherol had a 72% lower risk of ovarian cancer compared to women with the lowest intake of the nutrient. Some studies have examined the link between omega-3 fatty acid consumption and endometrial cancer risk. In one such study on 556 women with endometrial cancer and 533 healthy controls, a greater consumption of the omega-3s EPA and DHA, which are abundant in fatty cold water fish, were associated with significant lower risk levels for endometrial cancer. Specifically, women whose EPA consumption fell within the top quarter of the distribution had a 43% lower risk of endometrial cancer compared to women whose consumption was within the lowest quarter. Similarly, those who were consuming the most DHA had a 36% lower risk compared to those who were consuming the least. In addition, having a higher dietary ratio of omega-3 to omega-6 fatty acids has also been associated with a reduced risk. Finally, the women who consumed fish oil supplements had a 37% lower risk of endometrial cancer. Another study involving over 3,500 women found that women whose consumption of fatty fish, which are rich in omega-3 fatty acids, fell into the highest quarter of distribution, and they had a 40% lower risk of endometrial cancer compared to women whose consumption ranged within the lowest quarter of distribution. Omega-3 fatty acids like EPA and DHA may prevent cancer development through multiple mechanisms. These may include changes in the activity of gene expression and estrogen metabolism, as well as improved insulin sensitivity and reduced inflammation. Selenium is an essential micronutrient and is required for numerous metabolic processes throughout the body. Studies have shown selenium can disrupt estrogen signaling in cancer cells. Not only has selenium been shown to slow tumor growth, but it also decreases the risk of developing a variety of gynecological cancers, such as cancer of the uterus and the cervix. In 2009, a randomized prospective clinical trial showed sodium selenate to be beneficial for patients with cervical and uterine cancer who have a selenium deficiency and radiotherapy-induced diarrhea. In addition, a lab study on cervical cancer reported sodium selenate induces the death of cancer cells by apoptosis. Calcium is an important mineral and it's involved in hormone signaling, muscle contraction and bone health. While calcium plays a variety of roles in cellular signaling, it acts as a critical messenger in protein kinase signaling. The protein kinase C signaling controls a variety of pathways that are related to cellular growth and the regulation of cellular death. Calcium also plays a role in several other metabolic pathways that are related to cellular differentiation and proliferation, and these must be carefully regulated in order to avoid cancer. Women taking calcium supplements or who consumed calcium-rich foods were shown to have a significant reduction in the risk of developing endometrial cancer. Lignans are a group of natural phytoestrogens and they're found in plants like flaxseed and sesame. After consumption, the lignans can be metabolized into enterolactone, a compound that promotes cancer cell death, and it decreases the capacity of hormone-responsive cancer cells to grow new blood vessels to facilitate the tumor growth. While several studies are currently aimed at determining how the enterolactone might promote the endometrial cancer cell death, it's been postulated the phytoestrogens might compete with the endogenous estrogen for binding to the estrogen receptor. Given the estrogen dependence of the endometrial cancer, this hypothesis is consistent with the studies showing that women who consume high amounts of lignans have a 32% lower risk of developing uterine cancer. In postmenopausal women, this risk was 43% lower. Soy isoflavones are a class of plant phytochemicals and are found in soy and other legumes. A greater intake of the isoflavones are associated with reduced endometrial cancer risk. The soy isoflavones bind to the estrogen receptor and they modulate the estrogen signaling. Thus, they can act in a similar manner to the lignans to compete with the endogenous estrogen, which exerts more pronounced estrogenic activity. In 2011, a clinical study of postmenopausal women found those who consumed the higher amounts of soy isoflavones and total isoflavones were significantly less likely to develop endometrial cancer. And additionally, data from several case-controlled studies showed that soy and legume consumption was associated with a lower risk of developing endometrial cancer. For more on herbs, supplements, and natural treatment plans, check out my website.